This is Chapter 3, Cells of Living Units, Part 2, the Organelles. So to quickly review, in the last lecture we had looked at the plasma membrane, talking about the different functions of proteins within the plasma membrane, including intercellular joining and membrane transport. Today we will return to this general cell model, and we're going to discuss the different components of the cell, including the cytoplasmic organelles. When we discuss the cytoplasm, please remember anytime you see cyto to be thinking about the cell. So the term cytoplasm, this is a general term for all of the material between the plasma membrane and the nucleus. This contains the cytosol, which is the solution of the cell, and this is largely water-based, or aqueous, with dissolved proteins, salts, sugars, and some other solutes. We also have cytoplasmic organelles. The cytoplasmic organelles, these are going to be the metabolic machinery of the cell. We will talk about these at length. We will look at both membranous and non-membranous organelles. We also have things that are included called inclusions. These would be different chemical substances that we would find within the cell, including things like pigments, glycogen granules, and what are called glycosomes. Now, in terms of looking at the cytoplasmic organelles, these are specialized cellular compartments. You can think about these as being analogous to the organs in human systems. The two main kinds, or two main groupings we have, would be membranous and non-membranous. Membranous are bound by membranes. We will see examples would include the mitochondria, peroxisomes and lysosomes, the endoplasmic reticulum, and the Golgi apparatus. Whereas the non-membranous are going to be things like cytoskeleton, centrioles, and ribosomes. And these non-membranous will be discussed as we discuss the rest of the organelles. So the first organelle that we will mention is the mitochondria. In our general model, it's shown as these roughly kidney-shaped organelles rather. These have a double membrane with a shelf-like structure. These are called cristae. This is where we see aerobic cellular respiration occurring, and this is where ATP is produced. If you were to look closely, this is a photomicrograph of a mitochondria. You can see these shelf-like appearance of the cristae. Mitochondria, interestingly enough, have their own DNA and RNA, and this allows them to be a source of something called mitochondrial DNA. These are heavily laden with enzymes, and these enzymes allow for the production of ATP. So we can think of the mitochondria as the powerhouses of the cell. Now the ribosomes, these are non-membranous. These are granules that contain protein and what's called rRNA, or ribosomal RNA. These are going to be little machines that are going to allow for the protein synthesis to occur, and we will discuss how protein synthesis occurs later in Chapter 3 when we look at transcription and translation. Now, there are two different kinds of ribosomes. We have free ribosomes. These free ribosomes are going to be floating within the cytoplasm, right? And these are going to synthesize proteins that are soluble that we would see within the cytosol. Now, there are many that we will see as membrane-bound ribosomes, and these are going to be bound to what's called the rough endoplasmic reticulum. This is where we will see proteins that are going to be incorporated into membranes being found. Now, ribosomes have two main subunits. They have a small and a large subunit, and those have to come together to create a functional ribosome. Those membrane-bound ribosomes are found on what's called the endoplasmic reticulum, specifically the rough endoplasmic reticulum. Right? The endoplasmic reticulum is a continuous structure. Right? Here we have interconnected tubes and parallel membranes of the endoplasmic reticulum. These membranes are actually continuous with the nuclear membrane, the membrane of the nucleus, as we will see later. So here we have a section of the nuclear envelope, or the nuclear membrane, and here would be the rough ER and the smooth ER. Now, the rough ER is called rough because it does have embedded within it, it has ribosomes. The smooth ER lacks those ribosomes. It's also more tube-like as opposed to shelf-like. In the rough ER, we see the manufacture of all of the secreted proteins. This is where we are going to talk about translation occurring. okay, which we'll see later in the chapter. In a quick overview of how protein synthesis occurs. Protein synthesis occurs when the ribosomes are able to read directions on something called mRNA. And mRNA is going to be discussed later in the chapter. But take it in a simple sense that mRNA basically is like a recipe, and that recipe tells the ribosome how to make a protein. These ribosomes are all along the endoplasmic reticulum, so this would be the inside of the fold of the endoplasmic reticulum. This ribosome complex is going to basically have on it something called a signal recognition particle, and that's going to get this whole system started. Okay. As this ribosome works its way along the mRNA, reading the mRNA and creating the protein represented here by these little dots, those proteins are going to be pushed into this open space called the cisternae within the rough end. This protein will be pushed into this space called the cisternae. Right. As the protein terminates, is done being made, it then has certain sugars that are added, and eventually it will get folded into a three-dimensional conformation. If you remember from chapter 2, how proteins function is often due to their three-dimensional shape. Okay. It then gets enclosed in a transport vesicle. So earlier we mentioned vesicular transport, okay, or vesicular trafficking. These proteins, a little section of the plasma membrane of the rough ER, will bud off, containing within it the protein. That's then going to make its way to the Golgi apparatus, where we'll see packaging occurring. Now, the smooth ER is slightly different. The smooth ER, we do not see ribosomes associated with it. So instead of seeing protein production, we're actually going to see lipid production here. Now, how this occurs is going to be pretty different depending on where and what kind of cell we're talking about. So in places like the liver, we're going to see the breakdown of glycogen occurring and cholesterol metabolism. In the testes, we're going to see the synthesis of the sex steroid hormones. In skeletal and cardiac muscle, the smooth ER is going to be important in the storage and release of calcium. So the smooth ER is going to have very different jobs based on the cell in which it is found. Now, from this rough endoplasmic reticulum, when the proteins are made, those then get transported to what's called the Golgi apparatus. So here we see, shown in blue, a transport vesicle from the rough ER. It is going to be received on what's called the cis face, or the receiving side of the Golgi apparatus. The protein contained in this vesicle will be released into the space of the Golgi apparatus. It will then pass through. Here in the Golgi, we are going to see modification, concentration, and packaging of proteins. Okay. When that occurs, we're going to see that the proteins, depending on where they're destined to go, may be packaged in different ways. As these move through, we may see additional folding, additional groups added, okay, and then we're going to see these being shipped out, if you will, from the trans phase. Okay. So the different pathways that might occur, what may be going on is we may be producing here in the rough endoplasmic reticulum a protein that needs to be released from the cell, okay, or secreted. So it would be packaged into a transport vesicle, which would then be brought to the plasma membrane for it to be exocytosed. So here we have secretion by exocytosis. The protein may actually be a protein that is embedded within the plasma membrane. In this case, the vesicle has the protein within it in something called a codomer. That then gets incorporated in the plasma membrane, and it stays there, and we've now recycled, if you will, the proteins within our plasma membrane. A third pathway would be to create proteins that are going to be doing a job within the cell. One example would be enzymes that are going to be important in digestion that are called acid hydrolases. So in this case, we have a special kind of vesicle called a lysosome, and those lysosomes can carry these digestive enzymes or acid hydrolases to something called the phagosome. Right? You can also think of this as more or less the stomach of a cell where digestion can occur. So we might see any of these three occurring. Again, it would be highly dependent on what cell, what type of cell specifically we are talking about. Now, I've mentioned the lysosomes before. Lysosomes are important. These contain digestive enzymes, and depending on what cell type we're talking about, they may be digesting things that are ingested by the cell, like bacteria, viruses, toxins. They also may break down non-functional organelles. And interestingly enough, they are involved sometimes in the breakdown of glycogen and the release of thyroid hormone in particular cells. Now, if you've looked at this, we've seen that the endoplasmic reticulum and the Golgi apparatus are connected. They are physically connected to the nuclear envelope, and also the plasma membrane. So we can see that this is an endomembrane system, or a within membrane system. The overall goal of this system is to produce, store, and export biological molecules, and to break down harmful substances. It includes the nuclear envelope, the smooth and rough endoplasmic reticulum, those lysosomes, vacuoles, which are special kinds of transport vesicles, the Golgi apparatus, and the plasma membrane. So this gives us a connectivity. When we talk about the human body, we talk about systems. Here we see a system within the cell, okay? And this system functions. So you can imagine that a portion of plasma membrane may originate anywhere in this system and get slowly moved and recycled throughout the system. Now, shifting gears away from the membranous organelles, let's talk a little bit about the non-membranous organelles. Okay. First, we need to talk about the cytoskeleton, and this is quote-unquote the skeleton of the cell. Here we're going to see that we have this series of rods that run through the cytosol. We will see a high degree of variation in the type of cell and the type of skeleton they have. So the cytoskeleton is really going to be dependent on what the cell does and how it needs to move. Okay. The main types of, we will see that these are going to be proteins, and the three main types that we will talk about are microtubules, microfilaments, and intermediate filaments. So microtubules are made of a protein called tubulin. That tubulin subunit creates basically a spiral. These are approximately 25 nanometers. A nanometer is 1/1000th of a millimeter. Okay, so these are far beyond what you can see with the natural eye. Here we have a staining that shows you the nucleus in blue, and then these would be the microtubules surrounding it. These give the overall shape of the cell and the distribution of the organelles. Now we also have something called microfilaments. These microfilaments have actin subunits. These are going to be more dynamic. These are going to allow for movement. These will attach to the cytoplasmic side of the plasma membrane. Here we have a much smaller. These are 7 nanometers as opposed to the microtubules, which were 25 nanometers. Again, we see a staining technique here. We can see that there are much more of these microfilaments. The last group then are intermediate filaments. These are intermediate in size. These are approximately 10 nanometers. But rather than being globular proteins like actin and tubulin, here we have fibrous subunits. So we have these really strong, tough, insoluble protein fibers at a very high tensile strength. And you can see they wrap around another, wrap around and wrap around. So this is the same way you might see like a nylon hiking rope or a nylon climbing rope, where you have multiple subunits that are all twisted together to create a very strong. These we would find in desmosomes, which we saw in the previous lecture. These are going to help resist pulling and pushing forces. Now, another important component besides the cytoskeleton being is that there is a dynamic structure to the cytoskeleton. So cells can actually move, and they can move things within them. So when those vesicles we see going to exocytosis, they don't just float. Okay? Most of these are actually moved around using motor molecules. These are protein complexes that help to have motility within the cell. They utilize energy in the form of ATP, and these attach to receptors on organelles. Okay? So you can imagine we may have an organelle within the cell. Here is a microtubule of the cytoskeleton. Right? There we have what's called a motor molecule. This is going to use ATP, and it's going to actually change its shape so that this can move along the microtubule cytoskeleton. And it allows for there to be a directionality and an organization to how organelles may move. This can also occur in terms of moving parts of the cell or actually moving the cell. So you might see movement of skeletal elements, microtubules or microfilaments, relative to others, allowing for things like contraction of the cell and actual movement of the cell. So again, we have these motor molecules. Here they're depicted, they almost look like little feet walking along, and that's kind of how you can imagine these functioning. These proteins, when they take in ATP, they change shape, so you can imagine them almost being little feet walking along the cytoskeletal elements. Another important non-membranous organelle are the centrioles. Now, centrioles are going to be important in a few different cases. The most important is going to be during mitotic division, organizing the mitotic spindle, which we'll see in the next part of chapter 3. These centrioles, these are going to be pinwheel shaped. They're arrays of 9 triplets of microtubules. In cells, we will see these being a barrel-shaped organelle, and they're usually located near the nucleus. These are also involved in cilia and flagella, which we'll look at briefly. So here in our general cell model, here would be a centriole. Those centrioles are going to divide and move towards opposite sides of the cell in mitosis, and we will see that. Here you can see how they have a pinwheel shape. Each of these tubes that creates them, that is a microtubule. Okay, so these would be 9 triplets. So here's a triplet, 1, 2, 3, 4, 5, 6, 7, 8, 9. That's how we say they're 9 triplets. Okay. These centrioles, when they move to opposite sides of the cell, are going to be important in helping us divide chromosomes, which will be the goal of mitosis, which we'll discuss later. We also see these within cilia. Now, cilia are whip-like extensions of the cell that are exposed to the surface only on certain kinds of cells. These help to move substances across the cell in a single direction. And the way these relate to centrioles is the basal body, the part that actually hooks the cilia in place, is a centriole. The cilia itself is made of microtubules and these other proteins called dynein. Here we see the plasma membrane surrounds the cilium. The way a cilia moves, the cilia act like little whips. So they have a power stroke in which they move forward, and then a recovery stroke where they move back. Okay? And when you have not just a single cilia, right, but when you have many cilia together, as these beat and move, they are able to move substances. So this is going to be extremely important in our respiratory system. We will see that we have ciliated cells in our respiratory system, and that allows the movement of a layer of mucus along the cells. These are a certain type of cell called columnar cells, which we will see in chapter 4 when we look at tissues. So as these cells beat, they're allowing this layer of mucus to move. Right? So that's the main job. In terms of flagella, we don't really talk too much about flagella. The only flagellated cell in humans that has a flagella are sperm. Okay? We do see cilia in certain places, but we do not see flagella in any cell in humans other than sperm.